Thanks for the opportunity to present today, and also thank everyone for staying on a Friday, sunny Friday afternoon to listen to some more presentations. This presentation about photoacoustic deep tissue imaging will be taken from a previous talk, zooming out by a couple of magnitudes from the microscopic structure to imaging the entire animal. And I'm talking specifically about rodents, all with translation towards clinical application. So the talk is photoacoustic for deep tissue imaging. I took the liberty to modify it and add ultrasound. It's photoacoustic and ultrasound for deep tissue imaging. The main reason is that photoacoustic gives excellent molecular and function molecular information. Combining it with ultrasound for multimodality imaging gives also the anatomical location, the precise anatomical location in 3D, also 4D actually right now. So this can be used for multiple applications, and I by far will not go through all of them. If any of those keywords you see here sound very interesting to you, then please drop me a line. Happy to talk more about it. Also, the technology is featured in a number of big European grants, and you can see here on the slide. So for those who are not familiar with ultrasound and photoacoustic, or a combination of ultrasound and photoacoustic, some very brief animation. When ultrasound sends sound waves into tissue, and ultrasound I'm talking here is dedicated for small animals. So based on density, you get a reflection. And you get a reflection, you can do this in 2D, 3D, or 4 dimensions, where the 4th dimension is the time. An example here would be a mouse kidney. You see the kidney here in a typical ultrasound grayscale. Now, by having this dedicated transducer, you can modify it and attach fiber optic cables for laser light. It's pulsed laser, it's safe, it's diagnostic, not therapeutic. And by using laser pulses, nanosecond pulses, you can then use optical properties of the tissue. And Andreas had some of the photoacoustics examples from his side already in the presentation. So based on the absorbance, a sound wave is created that this transducer is detecting. And in that case, the red signal represents blood. So this is the kidney blood content. Combining different wavelengths, giving you then a so-called spectral mixing, allows you to do more advanced molecular imaging. For example, looking into oxygenation, like tissue hypoxia, how tumors respond, photodynamic therapy, normalization of vasculature, hemorrhage, thrombosis. All of these things can then be assessed by the optical properties. And those are done in parallel. So what you have here are optical absorption curves of hemoglobin as one of the nice endogenous contrast agents. And in principle, you can detect any contrast agent as long as it absorbs in a proper wavelength range. And proper wavelength for deep tissue is a near infrared range, because only the near infrared can actually penetrate this tissue below, beyond a few millimeters. And what I'm going to show you is deep tissue imaging going beyond the centimeter. The wavelengths that are discussed in this presentation are highlighted here in the boxes. So you have between 680 and 970 nanometers and 1200 to 2000, giving you good potential for a number of contrast agents, also clinically approved one like ICG, methylene blue, but also for imaging fat, brown adipose tissue, for example, one of those things that can be nicely imaged in vivo and in real time. So oxygen curve, I mentioned, oxygenation measurement correlate to a bolt. MRI, for example, done with photoacoustics also correlate to histology, which is also a key thing I'd like to highlight is the in vivo imaging capabilities can help in guiding the histology, where to embed the tissue, how to embed the tissue, and where to slice organs, tumors, etc. Deep tissue, meaning going beyond a few millimeters, can be done, for example, on the brain. So this advanced imaging and advanced processing. This is a completely non-invasive image. So this is a mouse brain. The skull is intact. And just by using the optical properties of the blood, creating this blood map or perfusion map on the mouse brain, allowing them to do several studies. Like in that specific case, they looked into epilepsy, but they can also look into strokes, stroke models, for example, anything that affects the vasculature, or stimulate the brain to see how the vasculature changes. Completely non-invasive. This is endogenous contrast, the blood. Several exogenous contrast agents exist, and for the sake of time, I'm going to focus on one specific. It's called Angiostem 800. This Angiostem is a near-infrared dye. It has fluorescence and photoacoustic imaging properties, and it's targeted against alpha V beta 3 integrins, which happen up here, vasculatures, also a number of tumors. There's several studies published with this already. And if you'd like to know more about it, it's a group at Optimal in Grenoble who work on specifically create this contrast agent. So if you want to then image the entire mouse, for example, imaging penetrating light from one end, light scatters, and you will not always reach a centimeter. So by changing the light delivery, illuminating the mouse from both sides, allows you to see the entire animal, in that case the angiostem, which is naturally accumulating in the kidney. Nicely seeing the contrast in the renal cortex. And next to it, you see the ultrasound image of the mouse. The spine, so this is the back of the animal, 1 and 2 kidneys, and you have a nice molecular information of the kidney in vivo, in real time, completely non-invasive. But now we know it's ending up in the kidney, but where else in the body can it be? And that's when ultrasound goes, and photoacoustic goes to image the entire body. So full body imaging, looking at biodistribution of a contrast agent. Is it in an organ? Is it going somewhere else? If you label drugs, for example, side effects, where do they go? Is the drug stable enough or stable long enough until it reaches the organ of interest or the tumor? All of this can then be assessed non-invasively by adding photoacoustic compatibility. And looking further into scanning the whole body, what the group in Grenoble did, endogenous accumulation in the kidney. So they fused the images with other imaging modalities, in that case CT, optical properties. Also then with photoacoustic being precise in localization of tumor metastasis in the bone. So these are bone metastases. Or glioblastoma tumors, non-invasively assessed in the brain, not touching the skull, you see it through skin and skull fully intact. All of this, great, you do this with animals. The overall goal, of course, is always to translate to the clinic. And to this end, a number of studies are ongoing currently under local ethical approval. One of the studies done is combining small animals and humans and looking in kidneys. For kidney transplantation, the state of the kidney in terms of how much collagen, how fibrotic the kidney is deciding on who should get an organ. So detecting collagen non-invasively in a fresh kidney is absolutely essential. So the specific study, they looked into the kidney of mice and were able to assess collagen, looked into large animals as well as human kidneys that are designed, dedicated for transplantation. And they were able to identify non-invasive with photoacoustics collagen content, which of course was then validated and verified. So they identified that using photoacoustic for deep tissue imaging have a way of tailoring organ transplantation. Nice translation story from mouse going to man. A number of other studies are ongoing under local ethical approval. I'll not go through all of them. Just really nicely to highlight what the researchers in Lund are doing using this ultrasound photoacoustic setup, the device you see here. So regular size ultrasound system. Here the tunable laser with cooling, everything included. Patient imaging, looking at the temporal artery for giant cell arteritis. And they're looking into vessel wall characteristics, molecular characterization of the vessel. And the overall goal is to avoid having to do a biopsy or to guide a biopsy rather than taking different samples to get an estimate of what's going on in the vessel, to know it non-invasively. Another endogenous contrast is melanin. And melanomas are a very frequent tumor, as we all know. There's a picture taken from a patient at the University of Galway, Ireland. The melanoma photography, but also looking with photoacoustics and the ultrasound in addition to it into where is the melanin. So the grayscale image gives you very good information about overall tumor, but then looking into the melanin, are the cells actually going through the basal membrane? How deep are they going? How deep are the lesions? That's specifically looked at with photoacoustic, non-invasively, and they also then combine this with 3D to get a parametric map and illustrating what the melanoma tumor looks like, also in terms of depth and penetration, tissue penetration. So all of this I showed you, I hope some of it was new. I hope some of it was already something that you have known. It's with a commercially available system. It's called the Vivo Laser X. If you'd like to know more, happy to talk to you about it. Full inherent co-registration of anatomical, functional, and molecular data in real time. All of this in a high-end ultrasound system with the laser next to it. Extended wavelength range, so really going to the near and far infrared. And with this entire setup being very flexible in light delivery, so you can really go deep tissue penetration. And also the data expert is versatile, so dedicated groups really work into the image processing and image analysis to even get more out of the images than what I could show you today. So the goal is in vivo imaging, non-invasive, to get molecular information, and subsequently at the end of an animal study to guide histology, like which section to take, how to embed the tissue, where to cut, which is also quite a significant time saver. And I hope I've used my 10 to 12 minutes. It was worth your time. And if there are any questions, I'm happy to answer them. Thank you very much.